Hello everyone, welcome to Biology Required Practical Microscopes. This comes up in two halves often. First of all, we've got preparing the slide. It's usually onion cells, but you often do it way back in year 9, so people have forgotten. And then the other bit of it is actually focusing the microscope. So we're going to cover both bits today to make sure that you are okay with both. Okay, you need to know the parts of the microscope. So if you have a look up here, these are the bits that we need to know. This is for everybody. Okay, I would rule out body tube. I've never seen that in an exam before. Eyepiece. This is our coarse adjustment. So our first adjustment to get it roughly in the right place. This is our fine adjustment. So once we've got it roughly in the right place, we can then get it totally in focus. The stage is the other one they ask about. Now this one's got a mirror, but sometimes they'll have a bulb. Okay, and this is the base. And then we've got these lenses. So the low power is the shorter of them. The high power is the longer. You've got your eyepiece up here at the top. And inside here is an eyepiece lens as well, and your objective lens. We'll be looking at those a little bit more in a minute. Now the equipment that you need other than the microscope is some onion, a slide, a cover slip, that's one of those tiny little squares of see-through plastic that go on the top, iodine solution, knife or scalpel, white tile and forceps. Now sometimes they ask us about safety, okay, and you think, well, it's a microscope, what safety have we got? However, have a look at our equipment. So we've got a scalpel, so we don't want to cut our fingers, so we're going to be cutting away from ourselves if they ask how could you reduce the risk. We have got, what else we got? Forceps, they're like tweezers. So again, be using them away from you because you don't want to like poke yourself in the eye with them or something. The microscope is very heavy, so we need to make sure it's right in the center of a desk so that it's not falling off onto our feet. And the last one, which is the bulb, can get hot, so do not touch the bulb even if you've switched it off, because it will stay hot for some time. So just be aware they might ask you about safety. And then we're going to take our onion tissue and we're going to peel off the tiniest or thinnest piece that we can. We want the cells as thin as possible, because if they are thick, the light won't be able to get through, and if the light can't get through, we won't see anything. Okay, so I'm just going to be adding bits on here, thin to let light through. Good. Now you're going to put that on the slide, flat down like that onto this slide. Now we're going to add some iodine. Now iodine is a brown liquid, and we do that to stain the cell walls or cell membranes so that we can actually see them properly. Now it says here, carefully lower a cover slip onto the slide. Now that's those tiny little square things. Now when it says carefully, what it means is you put one side down and then you put the other side down. So how could we put that? Let's see. Carefully lower a cover slip onto the slide, one side first. And what this does is it eliminates any air bubbles. Okay, let's put that over there. Eliminates air bubbles. They often ask you that. Okay, right, now it's time. Oh, hang on, there's another one we can pop in here. Sometimes when you put the cover slip on, the liquid squeezes out the side. So dab with paper towel I'm just popping down everything they may ask you. So why would you use a paper towel? To mop up the excess liquid, the excess iodine. Right, put the microscope onto the stage, and most microscopes have some clips that you can clip it in place. And that's how we make up our slide. Now we've got the focusing of the microscope. So we're going to turn to the lowest power objective lens. So that's the lowest one of these, which is the shortest one, this one here. So we're going to do that first. Then we're going to use the coarse adjustment knob, which is this big one here, so that the end of the objective lens is almost touching the slide. So we're going to turn our coarse adjustment knob here, where are we? There we go, until this objective lens is almost touching the slide here. Now it won't actually touch, but if we get it really close, that's when we can start our focusing. So now you're going to look through the eyepiece. If you're left-handed, use your right eye. If you're right-handed, use your left eye. That won't be in the exam, that's just the way your eyes and your brain work. So we're looking through the eyepiece. Turn the coarse adjustment knob in the direction to move the stage down. Now some microscopes the stage goes down, and in some microscopes the objective lens goes up. So either way is fine. And then you're going to do this, turn the coarse adjustment knob until the cells come into focus. So what you should be seeing are cells. You should be seeing, so you should be able to see the cell membranes. So you should be seeing something like this. Fingers crossed. Okay, now everyone's eyes are different. So you're now going to use the fine adjustment knob, which is a smaller one up here, this one. And to bring the cells into a clear focus. So you're going to turn it to as clear as you can so that you can see all those lovely cell walls, cell membranes. And you are going to be on the lowest magnification at this point still. Okay, so that's the basic focusing. Now, if you want to focus in a higher magnification, which on the school ones is probably 40, we are then going to rotate to the higher power objective lens. Okay, and when we do that, we will then refocus. So we will do these stages from 6 to 8 again. And most light microscopes, which is what we're using, have got 3 different magnifications. So there is a possibility you can do that 3 times. So that's all the focusing and how we make up our slide. So what I'd like to do is just have a look at the next bit, which is drawing our observations. So with scientific drawing that's through a microscope, they will often give you a circle to draw it in. Not always. They might just say, this is a cell that was under the microscope, please draw it. Now the trick is, in nature we don't have, very rarely, dead straight lines, okay? So it's going to be drawn freehand. Don't get your rulers out. You're not going to have rows of cells with a ruler. And you must always give it a title. So, for example, you might have drawing of an onion cell, or a blood cell, or whatever it is they give you. You then need to write down the total magnification. So if you are on foundation, they will just give you the magnification as something like, I don't know, times 500 or times 50, whatever the magnification is. If you're on higher, the possibility is that you could get asked to calculate the total magnification. So that is the magnification of the objective lens. So that's the ones by the stage that rotate, times by the eyepiece lens, which is the one up by your eye. Kind of makes sense. So let's say the objective lens was 30 and the eyepiece lens was 10. Your total magnification, too close there, it would be 300. Okay, so be aware that you might have to calculate those things. Right, now drawing. So often they'll ask you to draw, or they'll give you something that someone else has drawn and say, Jane drew this of a blood cell. Pick out 2 improvements that Jane could make, or something like that. All right, now what we're looking for are lines where there's no gaps. Because if you think about an onion cell, in nature, it's likely that the cell membrane is going to be complete. Okay, and if you're shoddy at drawing, you might go something like that, or actually leave a gap. Okay, it has to meet if at all possible. We don't want any shading. Okay, so that's our second rule. So none of this shading in, giving it a shadow on this side, like this. Okay, we don't want any of that. And we also we want clean lines. We do not want these sort of lines, because if we do these sort of lines, the examiner's going to go, no. We want clean lines. We don't want any gaps there where there shouldn't be gaps, or overlaps where there shouldn't be overlaps. We generally don't use a ruler until it's time to label our cell. So let me get rid of this one at the top a minute, and let's just draw one. Now you might have many cells in view, but I'm just going to draw one. And you might see this, so I can't colour in so well. Now when I say no shading, I mean no trying to put shadows in. But if you see something dark in the middle, then you can put it in. So let's see. We would then label, so this bit we can use a ruler for the lines. Let's say that's the cell wall, and that's the nucleus. That's probably all you'll see on one of the school microscopes. Okay, things like ribosomes, mitochondria, chloroplasts, all these things that come in a cell called organelles, little organs, that means organelles. They are too small to see under the school microscopes, sometimes even light microscopes. Okay, so if you had an extremely powerful light microscope, you might be better off with an electron microscope. Okay, so that's drawing. Really important we know that. You could either be asked to draw something from a cell or from an image under the microscope, or like I said, you might be given a drawing and say what improvements could be made, or what have they missed out, something like that. Okay, so be aware of that one. Now, just going to go to the next slide. So, if you get a microscope required practical question, please read it twice. Okay, the reason being, people look and they go, microscopes, oh okay, yeah, I know how to do that. But be careful, because some ask how to make up the onion slide. Some ask how to focus the microscope. and some will be a mixture of the two. How to prepare the slide and focus the microscope. And if you get that, it's important you share the marks out. So maybe 3 for the first bit and 3 from the second bit. Don't do 6 points just from focusing the microscope. Okay? Right, just to give you a quick kind of rundown of the things you need to be mentioning. So if you get one on make up the onion slide, we need 6 points. So let's go for the keywords. If you want to navigate yourself, pause the video. If not, we'll do them now. Okay, so thin onion cell, slide, microscope slide, cover slip. Now, there are marks for equipment as well as method. So chuck in the equipment you can remember. Add iodine. Then add the cover slip. Then dab with paper towel. Then put on stage. And clip. And if you're being really clever, remember that cover slip you put down one side first and then the other. So you could say put on an angle and then push down, something like that. It's always a mark. Good, so that's what you could be focusing on if you get how to make up the slide. If you get how to make up the, just put a line around here. How to focus the microscope, this is what you need to mention. You need to, obviously you've got the slide on the stage already. So you're then going to lowest objective lens. You're going to use the coarse the coarse focusing knob to get it in view. Then the fine focusing knob. Then you're going to increase objective lens and then repeat fine and coarse, sorry, in the other way around, coarse and fine. Okay, so that's what you need to be talking about. If you've got a mixture of the two, then you're going to have a mixture of 2 of those. Quite often with this question here, on the mark scheme, you have a look and there's like 15 different marking points. So the chances of you hitting them are quite high if you've taken in what we've done today. So I would at this stage, because I'm going to be doing lots of these required practicals, get yourself a revision card. On one side, how to make up the onion slide, and on the other side, how to focus the microscope. Just the keywords, that's all you need, because when you get in the exam, you can then extend them. But don't forget, you don't need to write sentences and sentences. Examiners are looking for keywords. They're looking for these words that we've said here. Obviously, you couldn't say, let's say, a coarse focusing knob. Let's say that one, right? So you could write down, use the coarse focusing knob. You couldn't say, put the coarse focusing knob on your head or something. That's not going to work. But you can make up that little bit of sentence when you're in there. Right, I really hope that's helped you. Be prepared to either write a method or look at a method, say what they've done wrong, or look at a method and they might ask you questions about why have the onion cells got to be so thin, safety, things like that. Okay, well done everyone. I will see you on the next required practical. Take care.